Okay, so the next chapter, this is a big unit. The next unit's a big unit. So we're going to start it now. I don't know if we're necessarily going to finish up this unit before the end of the quarter. This test might happen next quarter. The test you're having next week will be your third test grade or fourth test grade? Third test grade. Typically try to get you 3 test project grades a quarter. So that's about it. So this could happen, but it might not happen until second quarter. The reason why it's a big chapter is because we're going to talk a little bit about just metabolism in general. Then we go into a little bit on enzymes, and how enzymes help to regulate metabolism. And then we do a little photosynthesis respiration. So all that kind of goes together. So it's a big unit. So what are we looking at? We're looking at ATP, right? We're looking at energetics. So how energy is used to carry out the chemical reactions, to power our metabolism. Our metabolism is simply the collection of all of our chemical reactions. So pretty straightforward. We're going to talk about enzymes. We all know enzymes help to do what? Speed up chemical reactions. How do they speed up chemical reactions? By lowering activation energy. So we'll talk about that. But we'll also go a little bit more detailed and try to give you an understanding of how they actually lower the activation energy. So we'll get a little bit better understanding of what enzymes actually do. We'll talk about enzymes in the catalytic cycle. That's just talking about the cycle that enzymes run through. They're not used out. They can continue kind of moving through and carrying out chemical reactions. And then we'll talk a little bit about just kind of enzyme efficiency. Like talk about how temperature, pH, how competitive inhibitors can all affect enzymes. And then we'll talk a little statistics with Mr. Yu, and we're done. Okay. So we all know we're going to talk at the beginning thermodynamics. Right? How many people, entropy, right? Delta G, spontaneous reactions, things like that. So we're going to look at a couple of equations. You don't need to know these equations. These will not be on a test. But they're simply just to kind of introduce spontaneous reactions. So we all know kind of the main law of thermodynamics is that we cannot create or destroy energy, right? You guys have a ton of it right now. So we can't create or destroy energy. These are just some jellyfish right here. I forget does anybody remember the name of the gene? What's the name of the gene, the fluorescent gene? GFP, green fluorescent protein. That's it. It's a GFP gene. So it was a Japanese scientist who was studying these jellyfish off the coast, and he ended up isolating the gene that caused them to glow in the dark, glow green. And that was probably back, I would say maybe, I'm guessing 90s. I'm not exactly sure of the date. Probably like 1990s, I would guess, maybe a little earlier than that. But it ended up being actually a very good indicator gene. So in this class before, we don't really do it now, we do it with insulin, but we've actually made glow-in-the-dark bacteria by simply inserting this gene into bacteria. They make glow-in-the-dark cats, they make glow-in-the-dark mice, right? It would have been a good costume. Yes? You could absolutely make a glow-in-the-dark human. It would not be a problem. The only problem is, in the process, you might lose a few embryos. There's some ethical issues there. Plus, that human doesn't really have a choice to become glow-in-the-dark. It's not like we can make you glow-in-the-dark. We have to make an embryo glow-in-the-dark. So therefore, that person would grow up glowing in the dark. And they wouldn't really have a choice in the matter. It's not like we could reverse it. So it wouldn't be a good thing, but we could absolutely do it. Not a big deal. We could clone humans at this point if we really wanted to. It's just a matter of ethical reasons why we don't, even though some scientists in some countries have claimed that they have already. Any case, so GFP, right? The main thing here is we're looking at energy, we're looking at light. We can't create or destroy energy, right? Nor can we create or destroy matter. So we're looking at metabolism. Metabolism, again, right, it's the collection of all the chemical reactions that we are. So it's all the chemical reactions of breaking down food to release ATP, to make ATP. It's also all the chemical reactions used to build up our muscles. When we look at metabolism, we typically look at metabolism in terms of pathways. Pathways are just a series of chemical reactions, with every chemical reaction being regulated by a specific enzyme. So we talk about metabolism, all the chemical reactions, and we also break it down into metabolic pathways. Some pathways are catabolic, some pathways are anabolic. The two chemical reactions shown, obviously in mitochondria, we're looking at respiration as being a catabolic process. We're looking at photosynthesis as an anabolic process. Catabolic processes do what with energy? Absorb or release energy? Right, mitochondria is going to release energy. It's going to take glucose, a larger molecule, and break it down into smaller energy units, ATP. In order for photosynthesis to occur, though, it's going to require energy. So catabolic processes are spontaneous reactions, right? Anabolic processes are not spontaneous. They require energy. How do I remember this? Right? Baseball players, football players, they're always taking what type of steroids? Anabolic steroids. So therefore, they're using the steroids to build muscle. So how do you remember anabolic, catabolic? That's kind of how I always remember the two. All right, just a quick thing. This isn't really a big thing, but we're talking about energy. So just different types of energies out there, right? We know we have kinetic energy. We know we have heat energy, potential energy, and really the energy that we're focused on now is chemical energy, right? The energy that's harnessed in bonds. So our bodies are going to convert chemical energy into most likely some type of kinetic energy. However, we are not 100% efficient. So therefore, when we do these reactions, when we're changing chemical energy into kinetic energy, we always lose what? Heat, right? We always lose heat. Heat is an inefficiency. We use the heat to keep us warm, right? But it is an inefficient process. All right, thermodynamics, right? We have a few laws of thermodynamics, but before we talk about, we just need to talk, in general about systems. There are two types of systems in thermodynamics. Could say three, but two are kind of the same. We have closed systems and we have open systems. Closed systems only allow energy to transfer between the system and the outside world. Open systems allow the transfer of energy and matter. What are we? Are we closed systems or are we open systems? Are we open or closed? We're open systems, right? We're absolutely open systems. We have to take in food. That food gives us energy. It also gives us building materials, matter. Matter. What's an example of a closed system? Matt, what do you think? What's an example of a closed system? Fissionable is an organism that's going to eat. So all organisms are open systems. Plants they're still open, because they're going to transfer matter, carbon dioxide, right, nutrients, minerals, things like that. We could create like a laboratory condition. So we could create like a vacuum or something as simple as a thermos. A thermos is pretty much kind of considered a closed system. You put hot coffee in a thermos. Right, the matter's not really exchanging. It's really just an exchange of energy. The reason why we know energy is exchanged because when you fill up the thermos, your coffee's not going to be hot for 3 weeks. Right? It's only going to last maybe a day. So there's energy there. The Earth is kind of considered a closed system. Now, meteorites hit the Earth, absolutely. But for the most part, it's just energy from sunlight. It's kind of the overall. So the Earth, thermoses, things like that could be considered closed systems. Here are our laws of thermodynamics. Don't let this slide freak you out. You do not need to know this slide. I'm just kind of highlighting some of the key ones. Just to kind of go through it. Energy cannot be created or destroyed. We know that. Neither can matter. The entry of the universe is always increasing. We've already kind of talked about that in terms of diffusion. And really, those are the only two laws we care about. We don't really care about the other ones. The last one is just saying at absolute zero, you would have zero entropy, zero disorders, perfect crystalline structure. But we don't really care about that in biology. First law of thermodynamics, energy cannot be created or destroyed. It can simply be transferred. So just think about some standard chemical transfers. Energy transfers, such as your car engine. The chemical gasoline burns, right? That chemical energy is releasing and changing chemical energy into kinetic energy. Those explosions are pumping your pistons are pumping your drive shaft are turning your wheels, the car moves forward. Gravitational, right, with a hydroelectric power, right? There's gravitational energy that's being transferred into electrical energy, right? The river is flowing downhill. It's passing through the hydroelectric dam. It's spilling some type of energy generator, Really just a magnet inside some wires. That's all it is. Nothing too complex. And it's generating electricity. Solar, it's pretty straightforward. Nuclear, battery, going from chemical to electrical. So just thinking of some energy transfers out there. Okay, so the second law of thermodynamics. You can read this slide on your own. I'm really just going to skip to the second slide. We've already talked about this, right? Organisms do not violate the second law of thermodynamics, right? We maintain low entropy, but we don't violate the law because the universe is still increasing in entropy. Right? We don't violate the law because in order to maintain our low entropy, we must constantly be taking in what? Lots of energy, lots of food, right? Think about your lifetime, right? Think about how many hot dogs, how many hamburgers, how many salads, how many pasta bowls. Like think about all the food that you have consumed in your lifetime, right? And all those materials have gone from complex states to disordered states. We've created a huge path of disorder behind us, okay? So we've increased entropy in our world. By doing that, we've maintained our low entropy. So we don't disobey the law of entropy at all, and that's just kind of highlighted here. So, here's kind of your first law of thermodynamics, right? In order to get energy, you must transform it. So going from chemical energy in the food supply to kinetic energy in order to power the system. And you can think about the heat loss. You can think about the carbon dioxide and the water that's released, right? When you're actually breaking down the glucose and the proteins and so forth. So we are increasing the entropy of this world as we live. You guys are so quiet today. Okay, so here we go. Here's our delta G, our equation to calculate free energy. This is the equation I said you do not need to, we're not going to do any calculations with this equation. I'm just kind of showing it to you, and then we'll talk about the one thing we really care about in this equation is simply delta G. Delta G is a measurement of free energy. So how much energy in the system that is available to do work, okay? If our delta G is less than 0, it's saying that there is available free energy, and then that system can be spontaneous and it could possibly do work. If our delta G is greater than 0, it is a non-spontaneous reaction. Other parts of the equation, right, we're looking at heat change, we're looking at thermal energy, we're looking at entropy as well. They all play off each other. We really don't care about it. All we care about is that delta G concept. So really, this is what we want to look at. So I'll give you a second here. We're looking at two reaction curves. A reaction curve on the left and one on the right. The one on the left is looking at respiration. The one on the right is looking at photosynthesis. First question, which one is a catabolic process, which one's an anabolic process? So catabolic is the first one. Does everyone agree? Yeah, and then anabolic would be the other one, right? Catabolic breaks things down, right? Releases energy. Anabolic puts things together, requires energy. All right, so what we're looking at here, we're looking at reaction curves, and I like redrawing it anyways, though. So we are simply looking at this. We're looking at the energy of our reactants compared to the energy of our products, right? The energy of our reactants is greater than the energy of our products. The differences in energy, this is what delta G is. In this case, delta G is going to be less than 0. Therefore, this has free energy. Can this reaction happen spontaneously? Absolutely, right? It's considered an exergonic reaction, and it's considered spontaneous. So it's an exergonic reaction, and it is spontaneous. However, we look at this reaction, the reverse. This is respiration, breaking glucose down into carbon dioxide and water. This is photosynthesis, taking carbon dioxide and water and building it into sugars, glucose. Going in the opposite direction. So here, my delta G is greater than 0. It is a positive delta G. This one requires energy, so it is endergonic, and it is non. What we mean by spontaneous is it does not require an energy input. There's no outside help needed for this chemical reaction to occur. Yes.
For respiration, don't you need an activation energy?
That's a whole nother ballgame. So spontaneous doesn't mean that it happens quick, it's explosive. It just means that there's no overall energy input for this reaction to occur. Now it's going to need enzymes. You're going to need to reach activation energies, which is a slightly different thing. But overall, comparing the starting point and the final point, there's going to be energy released. So if you reach that the activation energy, the reaction can occur. Even if we reach, I guess you can say it here as well. So this is the energy barrier. That's our E sub A, our activation energy. Yes, there needs to be a little bit of bump in energy, but it can happen spontaneously. Here's the thing: if you kind of take away an activation energy, and we drop this off, the curve would look like that. And what that means is we would never have glucose in our world because there would be no energy barrier to hold glucose. And it also means that if we ever form glucose, it would revert right back to its original products. So the activation energy is that little bit of energy barrier that's needed to get a chemical reaction started. Without it, reactants wouldn't exist. The other thing here is the activation energy plays no part with delta G. So this energy here has nothing to do with delta G. Delta G is simply the energy of the reactants compared to the energy of the products. Okay. So why is this important? Sorry, just highlighting again. Before we move on, we'll kind of cover the same topic just kind of a different look. Right, we're looking at an exergonic reaction. Instead of looking at curves, we're just looking at graphs. Right, this is exergonic because our delta G is less than 0. Our reactants have more energy than our products. Energy is going to be released. Energy is available to do work. With respiration, energy is available to form ATP. Here we have our endergonic reaction. Right, we need energy to convert our reactants into our products. All right, so why is this important? Well, this is how we carry out metabolism. These are the concepts that help drive or to help ATP do its job. So when we're talking about this, ATP, the breakdown of ATP into ADP is a very exergonic process. There's a lot of free energy involved with that process, which allows us to couple the exergonic process of ATP being broken down into ADP. We can use those processes to power other endergonic processes. It's known as coupling, energy coupling. So here's our ATP molecule, which we should be familiar with at this point. Just breaking it down, our ATP is broken down into ADP, right? With the transfer of the phosphate group, there's a transfer of energy, so the energy goes with it. And this is what it allows us to do. So just take a second here. Was this in the right sequence for you? It's cool. When I move slides around, it moves around with you guys, so that's good. Okay, so all we're looking at here, is energy coupling. Right? All we're looking at is trying to take glutamic acid, react it with ammonia to form glutamine. In order for this chemical reaction to occur, it is an anabolic process. It is a process that requires energy. There's a positive delta G of 3.4 kilocalories per mole, just a measurement of energy. Right? So it's not going to happen. Right? This is non-spontaneous. It requires energy. So how do we power this process? Well, we do an energy coupling. We couple the reaction to form glutamine with the reaction of breaking down ATP. So now glutamic acid is phosphorylated, it is energized. So we add a phosphate group onto it. We then allow glutamic acid with the phosphate group to react with ammonia to form our glutamine. And if we take a look at the numbers down the bottom, we can see that this process is now a spontaneous process when coupled together. It is an exergonic process. So we can see our delta G with the formation of glutamine is positive 3.4 kilocal per mole. The delta G of ATP breakdown is negative 7.3 kilocals per mole. Therefore, our net delta G ends up being 3.9 kilocals per mole. It's simply the concept of energy coupling, and it's simply the idea that we couple exergonic processes in order to power endergonic processes. That's it. We're done. That's all we're talking about today. Yep, absolutely.